Hello, good day everyone. Today we will discuss about genetics session 4. We will continue discussing the extensions to Mendel's laws. A cross between pure late blooming and pure early blooming pea plants results in a first filial generation that blooms in between the two extremes. This is just one of the many examples of incomplete dominance, in which the hybrid does not resemble either pure breeding parent. The first filial hybrids that differ from both parents often express a phenotype that is intermediate between those of the pure breeding parents. Thus, with incomplete dominance, neither parental allele is dominant or recessive to the other. Both contribute to the F1 or the first filial phenotype. Incomplete dominance, the first filial hybrid resembles neither pure breeding parent. The figure shows the pink flowers, which are the result of incomplete dominance. The Antherinum majus or snapdragons. In Figure A, color differences in these snapdragons reflect the activity of one pair of alleles. In Figure B, the first filial hybrids from a cross of pure breeding red and white strains of snapdragons have pink blossoms. Flower colors in the second filial generation appear in the ratio of 1 red, 2 pink, and 1 white. This ratio signifies that the alleles of a single gene determine these three colors. In many plant species, flower color serves as a striking example of incomplete dominance. With the tubular flowers of 4 o'clocks or the floret clusters of snapdragons, for instance, a cross between pure breeding red-flowered parents and pure breeding white yields hybrids with pink blossoms, as if a painter had mixed red and white pigments to get pink. If allowed to self-pollinate, the first filial pink blooming plants produce F2 progeny, bearing red, pink, and white flowers in a ratio of 1:2:1. This is the familiar genotypic ratio of an ordinary single-gene first filial self-cross. What is new is that because the heterozygotes look unlike either homozygote, the phenotypic ratios are an exact reflection of the genotypic ratios. Another extension to Mendel's laws is codominance. The first filial hybrid exhibits traits of both parents. A cross between pure breeding spotted lentils and pure breeding dotted lentils produces heterozygotes that are both spotted and dotted. These first filial generation hybrids illustrate a second significant departure from complete dominance. They look like both parents, which means that neither the spotted nor the dotted allele is dominant or recessive to the other, because both traits show up equally in the heterozygote's phenotype. The alleles are termed codominant. Self-pollination of the spotted or dotted first filial generation generates second filial progeny in the ratio of 1 spotted, 2 spotted or dotted, and 1 dotted. The Mendelian 1:2:1 ratio among these second filial progeny establishes that the spotted and dotted traits are determined by alternative alleles of a single gene. Once again, because the heterozygotes can be distinguished from both homozygotes, the phenotypic and genotypic ratios coincide. In humans, some of the complex membrane-anchored molecules that distinguish different types of red blood cells exhibit codominance. For example, one gene with alleles IA and B controls the presence of a sugar polymer that protrudes from the red blood cell membrane. The alternative alleles each encode a slightly different form of an enzyme that causes production of a slightly different form of the complex sugar. In heterozygous individuals, the red blood cells carry both this one and this one, which are determined sugars in their surface, whereas the cells of homozygous individuals displays the products of either A or B alone. Another extension to Mendel's laws is multiple alleles. A given gene may have more than two alleles. If a person with blood type A mates with a person having a blood type B, it is possible in some cases for the couple to have a child that is neither A nor B nor AB, but a fourth blood type called O. The reason, the gene for the ABO blood types has three alleles. We have 1, 2, and 3. In this figure, allele A gives rise to blood type A by specifying an enzyme that adds sugar A. For this allele, it results in blood type B by specifying an enzyme that adds sugar B. This allele does not produce a functional sugar-adding enzyme. Alleles A, B are both dominant to this one, and blood type O is therefore a result of homozygosity of allele. Note that in this figure that the phenotype can arise from two genotypes. It is either this one or this one. The same is true for the B blood type, which can be produced from this allele. But the combination of the two alleles generates blood type AB. We can draw several conclusions from these observations. First, as already stated, a given gene may have more than two alleles, or multiple alleles. In our example, the series of alleles is denoted 1, 2, and 3. Second, although the ABO blood group gene has 3 alleles, each person carries only 2 of the alternatives. 1, 2, this one, this one, this one. There are the 6 possible ABO genotypes because each individual carries no more than 2 alleles for each gene. No matter how many alleles there are in a series, Mendel's law of segregation remains intact. because in a sexually reproducing organism, the two alleles of a gene separate during gamete formation. Third, an allele is not inherently dominant or recessive. Its dominance or recessiveness is always relative to a second allele. In other words, dominance relations are unique to a pair of alleles. In our example, this one is completely dominant to O, but it is called dominant with B. Given this dominance, the 6 genotypes possible with A, B, and O generate 4 different phenotypes. The blood sugar A, B, AB, and O. With this background, you can understand how a type A and type B parent could produce a type O child. The parents must be this one and this one. And, of course, heterozygotes. And the child receives an O allele from each parent. A person whose cells carry only A molecules, for example, produces anti-B antibodies. B people manufacture anti-A antibodies. AB individuals make neither type of antibody, and O individuals produce both antibody A and antibody B. These antibodies cause coagulation of cells, displaying the foreign molecules. As a result, people with blood type O have historically been known as universal donors, because their red blood cells carry no surface molecules that will stimulate an antibody attack. In contrast, people with blood type AB are considered universal recipients. They make neither antibody A nor antibody B, which, if present, would target the surface molecules of incoming blood cells. How do the multiple alleles of an allelic series arise? Mutations are the source of new alleles. The answer is that chance alterations of the genetic material, known as mutations, arise from spontaneously in nature. Once they occur in gamete-producing cells, they are faithfully inherited. Mutations that have phenotypic consequences can be counted, and such counting reveals that they occur at low frequency. The frequency of gametes carrying a mutation in a particular gene varies anywhere from 1 in 10,000 to 1 in 1 million. This range exists because different genes have different mutation rates. Mutations make it possible to follow gene transmission. If, for example, a mutation specifies an alteration in an enzyme that normally produces yellow so that it now makes green, the new phenotype green will make it possible to recognize the new mutant allele. In fact, it takes at least 2 alleles, that is, some form of variation, to see the transmission of a gene. Thus, in segregation studies, geneticists can analyze only genes with variants. They have no way of following a gene that comes in only one form. If all peas were yellow, Mendel would not have been able to decipher the transmission patterns of the gene for the seed color trait. The allele frequencies and monomorphic genes. Because each organism carries 2 copies of every gene, you can calculate the number of copies of a gene in a given population by multiplying the number of individuals by 2. Each allele of the gene accounts for a percentage of the total number of gene copies, and that percentage is known as the allele frequency. The most common allele in a population is usually called the wild-type allele, often designated by a superscript plus sign. A rare allele in the same population is considered a mutant allele. A mutation is a newly arisen mutant allele. In mice, for example, one of the main genes determining coat color is the agouti gene. The wild-type allele produces fur with each hair having yellow and black bands. These bands blend together from a distance to give the appearance of dark gray or agouti. Researchers have identified in the laboratory 14 distinguishable mutant alleles for the agouti gene. One of these is recessive to the wild type and give rise to a black coat on the back and a yellow coat on the belly. Another is also recessive to A and produces a pure black coat. In nature, wild-type agouti survive to reproduce, while very few black-backed or pure black mutants do so because their dark coat makes it hard for them to evade the eyes of predators. As a result, A represent at a frequency of much more than 99% and is thus the only wild-type allele in mice for the agouti gene, a gene with only one common wild-type allele is monomorphic. The allele frequencies and polymorphic genes. In contrast, some genes have more than one common allele which makes them polymorphic. For example, in the ABO blood type system, all 3 alleles A, B, and I have appreciable frequencies in most human populations, although all 3 of these alleles can be considered to be wild type. Geneticists instead usually refer to the high-frequency alleles of a polymorphic gene as common variants. One gene may contribute to several characteristics. Mendel derived his laws from studies in which one gene determined one trait, but always the careful observer, he himself noted possible departures. In listing the traits selected for his pea experiments, he remarked that specific seed coat colors are always associated with specific flower colors. The phenomenon of a single gene determining a number of distinct and seemingly unrelated characteristics is known as Pleiotropy, because geneticists now know that each gene determines a specific protein and that each protein can have a cascade of effects on an organism. We can understand how pleiotropy arises. Among the Aboriginal Maori people of New Zealand, for example, many of the men developed respiratory problems and are also sterile. Researchers have found that the fault lies with the recessive allele of a single gene. The gene's normal dominant allele specifies a protein necessary for the action of cilia and flagella, both of which are hair-like structures extending from the surfaces of some cells. In men who are homozygous for the recessive allele, cilia that normally clear the airways fail to work effectively, and flagella that normally propel sperm fail to do their job. Thus, one gene determines a protein that indirectly affects both respiratory function and reproduction, because most proteins act in a variety of tissues and influence multiple biochemical processes. Mutations in almost any gene may have pleiotropic effects. The same genotype does not always produce the same phenotype. What do we mean by genotype and phenotype? When we talk about genotype, it refers to the two alleles inherited for a particular gene, and phenotype or the observable traits. In our discussion of gene interactions so far, we have looked at examples in which a genotype reliably fashions a particular phenotype, but this is not always what happens. Sometimes, a genotype is not expressed at all. That is, even though the genotype is present, the expected phenotype does not appear. Other times, the trait caused by a genotype is expressed to varying degrees or in a variety of ways in different individuals. Factors that alter the phenotypic expression of genotype include modifier genes, the environment in the broadest sense as defined earlier, and chance. Retinoblastoma, the most malignant form of eye cancer, arises from a dominant mutation of one gene, but only 75% of people who carry the mutant allele develop the disease. Geneticists use the term penetrance to describe how many members of population with a particular genotypes show the expected phenotype. Penetrance can be complete or 100% as in the traits that Mendel studied, or incomplete as in retinoblastoma. For retinoblastoma, the penetrance is 75%. In some people with retinoblastoma, only one eye is affected, while in other individuals with the phenotype, both eyes are diseased. Expressivity refers to the degree or intensity with which a particular genotype is expressed in a phenotype. Expressivity can be variable, as in retinoblastoma, or unvarying, as in pea color. As we will see, the incomplete penetrance and variable expressivity of retinoblastoma are the result of chance, but in other cases, it is modifier genes and or the environment that causes such variations in the appearance of. Not all genes that influence the appearance of a trait contribute equally to the phenotype. Major genes have large influence, while modifier genes have a more subtle secondary effect. Modifier genes alter the phenotypes produced by the alleles of other genes. There is no formal distinction between major and modifier genes. Rather, there is a continuum between the two, and the cutoff is arbitrary. Modifier genes influence the length of a mouse's tail. The mutant T allele of the tail length gene causes the shortening of the normally long wild-type tail, but not all mice carrying the T mutation have the same length tail. A comparison of several inbred lines points to modifier genes as the cause of this variable expressivity. In one inbred line, mice carrying the T mutation have tails that are approximately 75% as long as normal tails. In another inbred line, the tails are 50% normal length, and in a third line, the tails are only 10% as long as wild-type tails. Because all members of each inbred line grow the same length tail, no matter what the environment, for example, diet, cage, temperature, or bedding, geneticists conclude it is genes and not the environment or chance that determines the length of a mutant mouse's tails. Temperature is one element of the environment that can have a visible effect on phenotype. For example, temperature influences the unique coat color pattern of Siamese cats. These domestic felines are homozygous for one of the multiple alleles of a gene that encodes an enzyme catalyzing the production of the dark pigment, melanin. The form of the enzyme generated by the variant Siamese allele does not function at the cat's normal body temperature. It becomes active only at the lower temperatures found in the cat's extremities, where it promotes the production of melanin, which darkens the animal's ears, this one, nose, paws, and tail. The enzyme is thus temperature sensitive. Under the normal environmental conditions in temperate climates, the Siamese phenotype does not vary much in expressivity from one cat to another, but one can imagine the expression of a very different phenotype, no dark extremities, in equatorial deserts, where the ambient temperature is at or above normal body temperature. Temperature can also affect survivability. In one type of experimentally bred fruit fly, Drosophila melanogaster, some individuals develop and multiply normally at the temperatures between 18 degrees Celsius and 29 degrees Celsius. But if the thermometer climbs beyond that cutoff for a short time, they become reversibly paralyzed. And if the temperature remains high for more than a few hours, they die. These insects carry a temperature-sensitive allele of the Shibari gene, which encodes a protein essential for nerve cell transmission. This type of allele is known as a conditional lethal because it is lethal only under certain conditions. The range of temperatures under which the insects remain viable are permissive conditions. The lethal temperatures above that are restrictive conditions. Thus, at one temperature, the allele gives rise to a phenotype that is indistinguishable from the wild type, while at another temperature, the same allele generates a mutant phenotype. Flies with a wild-type Shibari allele are viable even at the higher temperature. The fact that some mutations are lethal only under certain conditions clearly illustrates that the environment can affect the penetrance of a phenotype. That is all for this session. See you in our next one. Stay safe and God bless everyone. Bye!